In this training video, we will follow a conversation between an established investigator, Dr. Tact, and an early-stage investigator, Dr. Green, on the importance of adopting best practices for data sharing using persistent identifiers, community-endorsed standards, and a publicly accessible repository. We will see how Dr. Tact convinces Dr. Green's research team of the benefits of following FAIR principles and incorporating these into their data management and sharing plan. Let's listen.
Dr. Tact, I just completed a draft 10 IH proposal on mouse research. We will be conducting a comparative genomic study across mice and rats, focusing on genes with high conservation in humans.
Congratulations, Dr. Green. Please tell me more about the proposed study.
The study aim is to provide a comprehensive RNA-seq data set across several tissues for mouse and rat. This will provide access to a normal gene expression atlas for other scientists to use. We will also compare the tissue-specific expression patterns between the mouse and rat for a subset of genes with high sequence conservation with respect to human.
Sounds very interesting.
I'd like to share some information I plan to include in my DMS plan with you. This is my first DMS plan, and I would like to know whether my draft addresses the relevant aspects for sharing data most effectively.
Of course, I'd be happy to help you with your DMS plan.
Thanks. We plan to compare and analyze mouse and rat genomic data using a bioinformatics pipeline we built. Files will be stored on our lab computer and on a dedicated cloud host, Box, for long-term storage.
I see. This is a good start, but I think you can apply more best practices to increase the fairness of the research data. Making data fair means making it findable, accessible, interoperable, and reusable for other researchers. Since FAIR and DMS plan elements are interrelated, it's likely to strengthen your DMS plan as well. Can you tell me more about the experiments you plan to run?
Of course. Let me bring in our postdoc, Dr. Muse, since she's responsible for capturing, annotating, and sharing all the data. She can also give us best practice tips from her previous research experience.
Dr. Green and Dr. Tact will now meet with Dr. Muse to discuss the research project in more detail.
Dr. Muse, we wanted to talk to you about the plan for data sharing in this project. Would you please elaborate on the research plan, in terms of the data that will be generated?
Of course. We've selected 10 tissues to study from 3 male bulb C mice and 3 male Fisher 344 rats. We will extract RNA from the 60 samples for sequencing. We've created an automated bioinformatics pipeline to process and analyze the sequence data.
That's great, Dr. Muse. And how are you planning to store the data? What software tools will you use for accessing and analyzing the data?
We plan to use several open-source software tools for analysis, such as StarAligner, FastP, DuPredar, and Hermes. We also plan to create FastQ files from the raw sequencing data and all metadata necessary to run the R code. However, I haven't decided on where all the data files will be deposited yet.
This is a great start with using open-source software tools. Perhaps you can reach out to the research librarian for more ideas on a repository option to store your data. This is really important to be thinking about now, before you collect your first piece of data.
Dr. Green and Dr. Muse will now meet with Mr. Reed, the librarian, to discuss potential data storage ideas.
Hi, Mr. Reed. Thanks for agreeing to meet with us. We are eager to learn more about FAIR best practices and data storage.
Dr. Green, I'm glad you reached out to us. Have you considered our university repository yet? It's always a great first place to start for persistently capturing all the data you generate. Well-implemented institutional repositories, like ours, have a strong tradition in describing resources, providing access and building collections, and providing support for the long-term stewardship of digital resources. We apply FAIR principles in managing our very own digital data.
We did start there, and will be using the university repository to store our general data, but I think we want our shared data in a public, domain-specific repository to the mouse genomics community.
That makes sense. NIH provides information to assist in selecting suitable repositories for scientific data. For example, Array Express is a useful repository for high-throughput functional genomics experiments. The Mouse Genome Informatics, or MGI, repository at the Jackson Laboratory is another option. It's an international database resource for the laboratory mouse that provides integrated genetic, genomic, and biological data to facilitate the study of human health and disease. Both repositories use persistent identifiers, community-endorsed standards for data and metadata, and are publicly accessible.
Mr. Reed, thanks for your timely advice. One lingering question. I know fairness helps science overall, but what's the benefit of implementing FAIR best practices to our research team now?
This approach will not only catalyze your research by rendering all your lab data human and machine readable, but also streamline submission to journals and accelerate research data exchange with your current collaborators. By following FAIR principles from the start, you help both to satisfy DMS plan requirements and also accelerate your own downstream work.
And other postdocs, like the new postdoc we have coming next month, will be able to readily understand the data and evolve the research, and the data will be compatible to pool and reanalyze with similar data shared by other researchers.
Thanks, Mr. Reed. This conversation has been so helpful.
Dr. Green and Dr. Muse will now show Dr. Tact their updated plan.
Dr. Tact, I've refined aspects of my DMS plan based on our discussions to align better with FAIR principles. To summarize, 60 total samples will be collected. 10 tissue samples from 3 male bulb C mice and 10 tissue samples from 3 male Fisher 344 rats. RNA will be sequenced on an Illumina NextSeq 2000 and the data will be analyzed using Star Aligner with their corresponding Ensemble 110 reference genomes, FastPeak, DuPRadar, a bioconductor R package, and Hermes. The raw sequencing data and metadata will be captured in FASTQ files with all metadata in an ISA tab format. All files will be deposited to MGI. What do you think about us including this in the DMS plan?
This is a great improvement, Dr. Green. With this plan, you and the scientific community both benefit. And while we're on the subject of DMS plans, remember that submitted DMS plans are not set in stone. These may be enhanced in the future as necessary.
Thank you, Dr. Tact. Now I can see how FAIR benefits us directly and other researchers as well.
In this video, we've seen how a conversation between a senior and a junior researcher can lead to the adoption of best practices for data sharing using FAIR principles. By capturing data and metadata using community-endorsed standards, making it publicly accessible, and preserving it for future use, researchers can accelerate their own research programs and also contribute to the overall progress of science.